The human genome is made up of 3 billion chemical base pairs. Scientists often need to isolate a very specific segment of DNA from within a vast amount of genetic material. Since this segment is just one tiny piece of the genome, they need many copies to have enough to work with. The polymerase chain reaction, or PCR, can generate 100 billion identical copies of a specific DNA sequence in a matter of hours. As the inventor of PCR explained, the technique requires no more than a test tube, a few simple reagents, and a source of heat. To perform a PCR reaction, you need DNA that has been extracted from cells. Because the purpose of PCR is to make more DNA copies, you don't need very much DNA to start the reaction. For example, you could extract DNA from a small sample of blood, skin, saliva, or even a hair follicle. Start by moving your extracted DNA into a special PCR tube. The PCR works by heating and cooling the solution over and over, so PCR tubes are designed for even heat distribution. Next, add primer 1 to the PCR tube. Primer is attached to sites in the DNA strand that are at either end of the segment you want to copy. They are powerful tools for copying very specific DNA sequences, since there are almost no chance they will target the wrong sites. Now add primer 2, which will attach to the second site. Add nucleotides to your PCR tube. Nucleotides are the A's, C's, G's, and T's that make up the DNA code. These genetic building blocks will be used to create billions of DNA copies. Lastly, you'll add DNA polymerase to the PCR tube. DNA polymerase molecules act like tiny machines that read the DNA code and then attach matching nucleotides to create DNA copies. This particular DNA polymerase has been specifically selected to withstand the high heat of a PCR reaction. Now that the PCR tube is filled with all the reaction components, you'll place it into the DNA thermal cycler. This machine can precisely heat and cool your tube at specific times during the next hour. These changes in temperature are crucial for making the reaction work. Inside your PCR tube, cycle 1 has begun. The thermal cycler heats up to 95 degrees Celsius. That's 203 degrees Fahrenheit, which is almost boiling. At this temperature, the DNA double helix separates, creating 2 single-stranded DNA molecules. The thermal cycler now changes the temperature to 72 degrees Celsius, which activates DNA polymerase. When DNA polymerase locates the primer attached to the single DNA strand, it begins to add complementary nucleotides onto the strand. It continues until it gets to the end of the strand and falls off. Cycle 1 is complete. The same 3 steps happen in cycle 2. The temperature is raised again to separate the DNA strands. As the temperature is raised again slightly to stimulate DNA polymerase to copy the strand, click the next button to begin the 3 cycles. During cycle 3, your desired products begin to appear. The two strands that begin with primer 1 and end with primer 2. These are DNA copies of just the segment of DNA you have targeted. Although there are only 2 desired fragments at this stage, as you will continue with the cycles, these products will quickly become the majority. At the end of cycle 4, you have 8 fragments that contain your only target sequence. Now you have 20 fragments that contain your only target sequence and only 10 longer length copies. Click the fast forward button to run more cycles. After 30 cycles, there are over 1 billion fragments that contain your target sequence and only 60 copies of the longer length molecules. You now have a solution of nearly pure target sequences.